So this week we're going to work our way through the Precautions lab, which if we were in person, it is the coolest lab ever. We're going to kind of do a digital version of it this year. So we'll have to talk about it and you'll have to watch a video on it, and we'll try to break it up into little pieces to do something with this week. This is our last content for the biotech. So we'll be having a test this week. Thursday probably is our best day because that's the day that we would all be in the same place at the same time. I'd hate for you to use your in-class day taking a test. So probably we're looking at a chapter 14 test on Thursday. I'm going to go through this pre-lab lecture. I think if I skip videos, we'll get through it. And then tonight you have the Bozeman Science Lab 6 walkthrough. So it will also go into the restriction enzyme and electrophoresis portion of the Lab 6. And I'll just be going through the P-Glow portion. P-Glow, the glowing protein. There is a worksheet that goes with the Bozeman Science. It's not terribly long. The video I think is only 8 minutes. So I have that set to be due tomorrow because I think I'll spend a good portion of your hour today talking about the green fluorescein protein. So we already talked about we needed that Taq polymerase for the PCR. And now we're going to take advantage of a gene that comes from Aquaria victoria, which is a green fluorescein jellyfish. So we're going to take that jellyfish protein and we're going to incorporate it into a plasmid, recognize the plasmid map here, a plasmid of E. coli. And with that, we're able to transfer the plasmid in order to express the green fluorescein protein. And so that's the goal of this lab, to show transformation, much like Griffith showed transformation between his rough and smooth. Well, he was working with E. coli too, wasn't he? No, Streptococcus. That's what he was working with. Okay, so some background on the experiment itself, and then we're going to go into the steps. And you need to know, like I said the other day, you need to know the steps of each of these labs and why you do what you do. Okay, so we're going to try to transform competent E. coli, which means, first of all, we have to be able to define competent, into a form of E. coli that is antibiotic and can express the GFP protein. Okay, so we need to thoroughly understand plasmid, which I think we do, and the GFP protein, the green fluorescein protein. So just a reminder of the central dogma, and I see I need to adjust my letter size. DNA is transcribed through RNA, which is then translated into proteins, and proteins are expressed as your physical traits. This is your phenotype. RNA, the genes that you have, remember that's your genotype. And we learned there's all kinds of different ways to control whether an RNA is expressed or not expressed. And then we have ways of altering proteins once they are created. So this is Aquaria victoria, which is a bioluminescent jellyfish, and they have a very interesting life history. If you want to read about them, you have the link to this PowerPoint. So you can click on any of these hyperlinks. That is a hyperlink. Color-coded surgery. I love watching this video in class. It's about 20 minutes. It's a TED Talk. The girl is fascinating, and she is talking about how we can use this green fluorescein protein to identify cancerous cells in humans, and therefore eliminate biopsies that we don't need, and to make sure that we get all of the tumor that we need, because sometimes they have little stragglers that you don't see with the, just with the naked eye. So if you're able to make those cancer cells glow, you're more likely to get all the bits and pieces, if you will. A lot of times they take biopsies and then they have to go back in and take more biopsies because they don't get the boundaries, and so they need to keep going until they get to the edges of that tumor. And with this color-coded surgery, you're able to get all of that. So that's an awesome video. I would definitely show it if we were spending an hour in class. But I feel like if you're going to watch videos, that's probably not what we should do with our Zoom time. So you can see that we're incorporating that green fluorescein protein here into the genome in order to use that as a tracer. Remember, we've talked about using radioactive isotopes as tracers. The heavy isotope allows you to follow a chemical through a chemical reaction. We can use the same thing with this tracer by tagging on that fluorescein protein. So for our transformation, we're trying to take E. coli that is not expressing a green fluorescent protein, and we want to turn it into one that does. So what we are going to need is, first we're going to need some bacteria, and we're going to grow them on a petri dish. So normally we would do a starter plate. And so that starter plate, we're going to streak plain E. coli. We would put it in an incubator overnight, which is like a little oven, and we would set it at 37 degrees. And so this is kind of optimal for E. coli's bacterial growth. So that would allow this somewhat invisible amount of bacteria to grow into a visible colony of bacteria. And then we're going to scrape off a small sample of that bacteria, and we're going to put it into 2 centrifuge tubes. So one, we're going to also add we're going to add the we're going to add the P-glow plasmid too. So we're going to actually label this tube plus P-glow. And in the other tube, we're going to label it minus P-glow. So this one will be our control. This is not going to get the plasmid. So we will put both of those on ice for about 15 minutes. Then we're going to shock them with heat for 2 minutes. And then we are going to incubate them at room temperature for 10 minutes. So that's the little centrifuge tube that has your bacterial colony in it after you have already grown it overnight, okay? We're going to streak them on 4 different plates. Notice some of them are labeled LB. Well, they're all labeled LB. LB is standing for Luria Broth, which is basically food for the bacteria. Some of them are labeled with AMP. This has ampicillin incorporated in the gel itself. Ampicillin is an antibiotic related to penicillin. And then this one is also going to have Arabinose, O-S-E ending. We know that this is a sugar. So this contains all 3 things: the broth, the ampicillin, and the Arabinose. So in order to get the bacteria to take up the plasmid, we're going to subject it to these different conditions, the heat and the cold. And we also need to use a set of salts to block the negative charge of the DNA. So to be competent means that the bacteria have the ability to take up that plasmid or other macromolecules that you might be trying to force in that bacteria. So the cell wall must become permeable. They normally do not express this competency. We can use chloride salts to increase the ability of the bacteria to take up the plasmids. And then also those temperature changes are going to aid in the process as well. So we want to know why we're using each of these different things, okay? So here we can see DNA in the external environment. This is the bacteria and its own DNA. And we want to shuttle this DNA into the bacteria, okay? Along with that DNA, we are going to shuttle in the GFP protein and the ampicillin resistance protein. So here is our plasmid, and we are going to incorporate our proteins, our protein gene and our ampicillin resistant gene into the Ori operon. So remember, we use plasmids quite often as vectors. In genetic engineering, we talked about wanting to grow insulin in the lab, for example, so we might incorporate that insulin into the plasmid. So that means we can use these plasmids to express all kinds of different genes, since every organism goes through the central dogma in the same way. Okay, so these circular DNA, normally, this is not the average genome of the bacteria. This is like superpowers or DNA they've picked up from other sources. So this isn't their main genome. And plasmids, you know, they replicate on their own as well. So here you can see the supercoiling of the plasmid. Okay, so let's keep going with that. So in order to make them competent, in order to go through conformation, we're going to use a very small portion, like 10 micrograms of this plasmid. So we're incorporating it into this this Luria Operon, which includes beta-lactamase and the ampicillin resistance and the GFP. Okay, so once this operon is turned on by the presence of arabinose, it'll express all structural proteins, all structural genes within that operon, right? Which means it's also going to express the green fluorescein protein and the ampicillin resistance, okay? So we already showed that. So the lactose operon, Arabinose Operon, so we've worked the lactose operon quite a lot, right? So we understand how that works. So the Arabinose Operon works in the same way. In the presence of arabinose, it will be turned on, right? So the arabinose works as a promoter, and it will bind with that operator to turn the operator on and allow polymerase. It binds with the regulatory gene, which then is removed from the operator, which turns the allows the polymerase to proceed. Okay. And again, I have links. This is the Arabinose Operon in specific. I think it is 4 minutes maybe, but it's kind of lecture style, but he'll explain the entire operon if you want to go into that in more detail. We've worked with the operon a lot, so I feel like we're pretty confident in it. This is the video you're going to watch tonight. This is showing you how to start a streaker, how to streak your plates so that you don't get a whole like lawn, the entire petri dish covered in bacteria. You want to kind of separate the bacteria out so that you can try to isolate individual colonies. So basically you would put your main sample in a dab here, and then you would draw, you have inoculating loops, which are skinny straw, like kind of like a coffee straw, you know, not like a drinking straw. And you would streak through that main sample, and then you're going to zigzag through your plate, which kind of spreads it out. And then you're going to cross that original spread, which further streaks. It reminds me of if you were trying to make like a turtle brownie. But anyways, it'll take a small sample of that original streak and spread it out further, which allows you to isolate individual colonies. Okay. So we're going to suspend our bacterial colonies in 250 microliters. We use these micropipetters that allow us to get very specific amounts, very small, specific amounts of different solutions and administer them. So we're going to take about 250 microliters, and I can show you the pipetters tomorrow when you come to class, of this transformation solution. And the transformation solution contains the calcium chloride. And we will put this transformation solution in both of our little centrifuge tubes, both the one with the plus picolo and the minus picolo. We're going to add the plasmid only to the plus picolo. We put them on ice. We will shock them for 90 seconds at 42 degrees Celsius. So I have a hot water bath that we would put these in at 42 degrees Celsius, and we leave it in for exactly 90 seconds. The timing and the temperature is very specific for this lab. And then we're going to place it on ice again for 2 minutes. And then overnight, we will, or not overnight, but for 30 minutes, we will incubate it at 37 degrees Celsius. This is a video clip on how to streak your starter plates. I'm going to show that video clip tomorrow. So we want to know why we're placing it on ice, why we're using this transformation fluid, why we are using the heat shock. So this is the transformation. So notice the negative charges on DNA. The phosphates make the DNA very negative. So the calcium portion of the salt will bind to those phosphates and block that negative charge. So the negative charge would have been repelled by the hydrophilic portion of the phospholipid bilayer, right? So we needed to block that charge. Then we're going to put it on ice and we're going to heat shock it. Why are we doing that? So we know that molecules move more slowly when temperatures are cold, right? Which means the phospholipids that are in constant motion will move more slowly. And why would that be a benefit? Because I have an opening for which my DNA can slide through. If it's moving constant like this really fast, not going to be very easy to get that DNA in, but notice the gaps that occur when it goes more slowly. And so the DNA is able to slip through. Okay. So the heat shock is making an opening. I'm sorry, the ice bath is making an opening. And then the heat shock is creating a current to assist the movement of that plasmid through the membrane. So it kind of sucks it through. The nutrient broth you're using simply as food for the bacterial, but also you're providing that sugar that it needs to turn that enzyme or to turn that plasmid on. Okay, so here's your Luria broth. This is what you find in it: carbohydrates, amino acids, nucleotides, salts, and vitamins. We would melt this. It starts out as a gel. This is a very labor-intensive lab. So you would come in after school one day and we would melt down this gel, and then we'd have to pour these streaker plates while they're still hot. So we would pour that in and then we let it set. We let it cure, if you will, for 2 to 3 days before we actually start the lab. So we're not going to be doing that. You're going to work with these volumetric pipetters tomorrow, and the group that comes in on Friday, I'll let you play with them as well. But these micropipetters have very specific increments. Okay, so remember, we're going to be using 250 microliters of our transformation fluid. So we would have to squeeze our pulpate bulb, put our pipette into the very small tube all the way to the bottom, and then we would release ever so slowly until we suck up just 250 microliters. We don't want to get any air in there. And then we would administer, squeeze it out, our 250 microliters into each of our pipetters. Okay, these are our labels for our Petri dishes: Luria Broth plus ampicillin, Luria Broth, ampicillin, Arabinose, and Luria Broth by itself. So the difference between a colony and a lawn grows over the entire Petri dish. You can't identify individual. It's not that you would identify individual bacteria, but you wouldn't identify like individual families of bacteria, whereas a colony would be considered that family, and they show up more like a polka dot. Okay, so can you guess on these Petri dishes, which one would all bacteria grow? Which one would you get a lawn on? And you can shout it out or you can put it in the chat. I'll open my chat. Which of these will bacteria grow on? So you can use the names or the numbers. All bacteria, a lawn, a complete coverage. Okay, so all of the bacteria will grow on the Luria broth only. Ampicillin is an antibiotic. It's going to kill off bacteria. So these two here will have only colonies, because not every single bacteria will take up the DNA. It's actually difficult for them to do that, right? So we're not going to have 100% transformation efficiency. So we need to get rid of the bacteria that don't express the gene we want. If we were making insulin, we want to get rid of the bacteria that aren't growing the insulin, because that bacteria will create an overgrowth and you won't have as many of your selected bacteria. That's the purpose of putting the ampicillin in there. It's a selective crusher. So this LB plate should grow all bacteria, transformed or not. These two, because of the ampicillin, should only express the bacteria or should only grow the bacteria that have the plasmid. So which of these two will create colonies that glow, 1 or 2? So both of them will grow bacteria that have transformed. But without the arabinose, the regulator protein maintains contact with that operator, and polymerase cannot transcribe the green fluorescein protein because it's incorporated into that operon. So we needed the arabinose to turn the operon on, which you guys got. Good job. So I probably should have had this slide before the other one so you could see the difference. So this is a lawn, and this is these are colonies. So we're trying to get colonies of growing proteins. And this was also the purpose of streaking our plate and trying to spread out the bacteria. I wanted to get individual colonies rather than a whole lawn. You see that these 4 discs look a little bit different. So these 4 discs are 4 different types. This is a totally different lab, but these are 4 different antibiotics. And this is showing you a zone of inhibition. You could measure this out. So these, so this is just showing you how the bacteria won't grow in the presence of that antibiotic. Okay, so in this lab, some precautions we would take is, 1, if we were doing the lab, I would ask you if you were allergic to penicillin, ampicillin, kanamycin, or tetracycline. If so, you would not be working with the ampicillin plates. No food or drinks in the lab area. Anytime you're working with bacteria, no food or drinks. You're going to wash your hands before and after the lab. So remember, we're trying not to bring in anything to our lab. We want as sterile a location as we can, and we don't want to take anything away with us. So often we just bleach down plates or our tables after. We would also bleach our tables before. We, of course, would wear goggles and gloves, and all of the supplies that we use, we would bleach before getting rid of. So we would throw these away in their own bag, so they'd be double bagged, and we'd bleach everything before we throw them away so that we're not introducing bacteria into our environment. All right. So without showing videos, I was able to go through that a little bit more quickly. What questions do you have?